Thank you for the introduction. And you've probably been wondering what you're staring at on the screen because there's a lot of cake, right? There's a lot of cake on the screen. There's cake brain, there's a cake heart, and cake lungs, and there's people eating cake in the lab, which you shouldn't do. But you're going to have to figure out what we're talking about with cake today and why I'm talking about cake. So let's get into it. So, what is our cake for today? Cake could be one of these lovely science-themed cakes. There's some DNA, there's a microscope, there's some beakers, but I'm going to define for you what our cake actually is today. So our cake is a dense, multi-layered scientific topic. Each layer represents different aspects or components of that topic, intricately designed, rich with flavor, much like how our scientific findings are packed with complex information and details. So that's our cake for today. Now, this talk is about science communication if you read the booklet. So I'm going to relate the two now, but first I need to define what science communication is. Science communication is the practice of conveying scientific information to diverse audiences in clear, engaging, understandable ways, with the ultimate goal to bridge the gap between scientists and the public to those that are not knowledgeable on particular subjects. That's our overall goal here. And so my question for you, which I'm going to go through today, is how does science communication deal with the cake that I just defined with you on the previous slide? These lovely other AI generated images. If you're wondering if these are from AI, I've gotten very good at manipulating AIs to make weird images. They probably think I'm obsessed with cake at this point. The amount of cake images I made it generate were way too many. So how does science communication deal with the cake? A science communicator, such as myself for you today, acts as a skilled baker who slices this towering cake into smaller bite-sized pieces. These manageable portions are easier to digest and enjoy, allowing everyone from seasoned connoisseurs to curious novices to appreciate and understand the cake's complexity without being overwhelmed. Okay, that's how science communication is dealing with our cake. But what is our cake for today? So I'm assuming there aren't that many microbiologists in the audience here. No, this is why I chose this topic for today. So we're going to be talking about an antibiotic produced by a bacteria who this lovely image depicts here. This is a bacterial colony from the Penibacillus species. And if you don't know, this is one of the species of bacteria that produce antibiotics. Streptomyces is another common bacterial type that produces antibiotics. But today, I'm talking about this specific bacteria because a newly discovered antibiotic was recently found. So, there was a Nature paper published recently in March 2025 titled A Broad Spectrum Lasopeptide Antibiotic Targeting the Bacterial Ribosome. That sounds complex and ambiguous, right? And I want you to focus on this select topic here in the quotes. The newly identified Lariocydin antibiotic inhibits bacterial ribosomal machinery at a new ribosomal site, resulting in inhibition of translocation elongation. Who knows what that means? Maybe someone who took biochemistry recently understands that there is translocation that occurs in translation when you make proteins. But we're going to break this down for you today so you can understand how this new antibiotic actually impacts the translation process. Okay, so how do we tackle this cake of Lariocydin, this new antibiotic? I need to be able to convey to you the key points necessary to understand the basics of how Lariocydin works. And how do I do this? I'm going to be discussing today the audience, that is you, the clear visual that you need to capture this, and clear vocabulary that it takes to depict a topic that might be complex or unfamiliar to an audience. And so who's your audience? You are my audience for today. You consist of a wide variety of specialties, a wide variety of, there are students, there are professors who have been in fields for a long time, who are very well versed in their topics, and students who may not be very familiar with a lot of core science topics yet because they haven't had classes. And so your audience might consist of a college student, high school student. We have master's students in the audience. We have PhD students in the audience. There's definitely professors in the audience here. Senior scientists, this usually talks about someone in an industry setting. We don't have any of those here, although I think Daniel used to be a senior scientist, right? We definitely have cardiovascular scientists here, and we definitely have neuroscientists here as well. And so, again, that's why I chose a microbiology topic, and I'm going to be talking about ribosomes today, because you probably don't have to think about that quite often if you're in these fields. And so I'm going to talk about how do you hold your audience's attention, and then I'm going to get down into breaking Valeriocydin into a bite-sized piece. So how do you hold your audience's attention? Pace is a really important thing to consider. You don't want to be too slow and boring. You don't want to be too fast and overwhelm someone with a stampede of words that they're going to completely stop listening to you. You need a strong hook. What's a hook? Well, it could be a question that you start off with to grab the audience's attention. It could be weird cake images on the beginning of your PowerPoint presentation. It's something that you can relate directly to your presentation that you're giving, and it grabs the audience's attention. Tell stories, be interactive if you can, poll the audience or grab details from the audience that you can use. Use analogies and metaphors. We're using cake today. Keep it concise and focused. You don't want to be going off onto tangents and lose the audience's attention. That's one way to lose the audience. Clearly labeled visuals are extremely key. Minimizing the amount of text to only what you need, and you want to either eliminate or clearly define jargon that might be unfamiliar to a topic. So, breaking down Valeriocydin for you today. We're going to talk about the cake, and we're going to talk about the bite-sized piece. So, the cake is this statement that I showed you before. The newly identified Valeriocydin antibiotic inhibits bacterial ribosomal machinery at a new ribosomal site, resulting in inhibition of translocation elongation. This is that expert statement that only people in this field typically would fully understand what's going on. So how might I change this statement for someone who is not familiar with this topic? I'm going to change it to this, a bite-sized piece. The new drug has been discovered called Valeriocydin. This drug prevents bacteria from being able to make proteins, which in turn prevents them from being able to replicate. Does that make it a little better? I hope so. So we're going to go down that route and go for explaining this topic to a student and not someone that's an expert in the field, because none of you are biochemists either, I think. All right, so clear visuals are extremely key, and people need to know key information in order to understand a topic. If we're talking about protein synthesis, we need to talk about ribosomes. And so ribosomes are organelles in your cells. And there are different types of ribosomes. There's prokaryotic ribosomes and there are eukaryotic ribosomes. Prokaryotic is in bacteria and eukaryotic is in like us. And so there are the prokaryotic ones, which are called 70S ribosomes, and eukaryotic, which are called 80S ribosomes. And they're made up of two components, a large subunit and a small subunit to make this large 70S complex or this 80S complex. And this is really important because we're talking about an antibiotic here. And so this antibiotic has to recognize something to be of use to stop protein synthesis. And so that's why I'm defining the pieces of those in the prokaryotic bacteria and those in eukaryotic like us. And so prokaryotes have a 50S large subunit and a 30S small subunit, which combine together again to form the 70S subunit. And one of the components of this prokaryotic ribosome is going to be the target of this antibiotic that I introduced to you. So this is our ribosome broken down. And so then next, what you need to understand is, what do we have here? Let's see how this works. We have the large ribosomal subunit complexed with the small ribosomal subunit. Great, and there's a couple other things you need to know about on this slide. We have mRNA, which holds the genetic code that gets read like a book to make proteins, and we have this tRNA over here, which seems to come and interact with the ribosomal subunit. And there are three sites within the ribosome, this A site, this P site, and this E site that this tRNA can exist in. What's the purpose of a tRNA? A tRNA recognizes specific codes on the mRNA and brings in a specific amino acid that gets put together to eventually make your full protein. And this is how protein synthesis occurs. More tRNA will come into this A site, move to the P site, go to the E site, and then get kicked out of the ribosome eventually creating, again, this long amino acid peptide to form our full protein. And so there is a reason I'm defining this, because these sites might be important for how the antibiotic is interacting with the ribosome and possibly the tRNA. And so how does this drug, this antibiotic, lariocydin, interact with the ribosome? Well, lariocydin, it's this lasso. It acts like a lasso and binds both the small subunit of the ribosome and the tRNA that sits in the A site. So if you'll see, this lasso here is depicted of lariocydin, and it's binding a portion of the small subunit and this tRNA that sits in this A site on the ribosomal complex. Great. This results in no protein synthesis because this prevents the ribosomes from being able to continue reading the mRNA and will halt the building of your protein. But why is this antibiotic specific for inhibiting prokaryotes if eukaryotes also have ribosomes? That's why I identified the different types of ribosomes before. Because lariocydin antibiotic specifically binds to only the 30S ribosomal subunit, which only exists in prokaryotes and is not found in eukaryotes. So this antibiotic will have no effect on humans, while it is good at blocking protein synthesis in bacteria and stopping the growth of bacteria, therefore. And so, why is this antibiotic different than others that we know of? Why is it different? Well, I'm going to bring up this statement again. This newly identified antibiotic inhibits bacterial ribosomal machinery at a new ribosomal site, resulting in inhibition of translocation elongation. So this indicates that the other antibiotics that we know of bind to different sites on the ribosome and inhibit protein synthesis in different ways. So this is a new site that gets targeted by this antibiotic. If you can look here, we have chloramphenicol, right? It binds to the 50S large subunit. We have tetracyclines. It binds to the 30S subunit just like lariocydin does, as well as aminoglycosides. But why is lariocydin different? Because lariocydin is the first antibiotic to bind to the 30S small subunit and the tRNA located in the A site of that ribosomal complex. And so why do we care about that? Why is that relevant? Well, have you heard of antibiotic resistance or bacterial resistance to treatment? This has been on the rise chronically for decades now and has become a severe problem because patients can't be treated. And we're running out of treatments for people who are actually infected by bacteria. And so bacterial resistance is a major problem. You can't treat your patients. And so this is a new antibiotic that might be able to be used for patients who are infected with bacteria that are resistant to these current treatments of antibiotics present on this slide here. So very critically important. And so maybe you're wondering where these graphics came from. These are some of the tools that I use in order to build graphics to make good figures. I made the ribosome figures using Canva. If you've heard of Canva before, it's very easy to use. It's free. You can pay for a subscription as well that gives you a cool AI tool where I made all the cakes on. But you can also make AI pictures if you use ChatGPT, for example. You know Adobe Illustrator, right? This is a paid subscription. It's typically really the gold standard. It's for making figures, but it's paid. So if you want a free version for making something, use Inkscape. Inkscape is basically a free version of Illustrator, allows you to make high resolution vector tools that you can blow up your images to large scale without them becoming pixelated. BioRender is a software that gives you 3 canvases for free, and it allows you to use clip art, that's scientific clip art that's made by scientists and illustrators. So you can create posters, presentations, and all the clip art is made for you, so you don't have to draw anything for yourself if you're not into that. SciDraw is also a free site where you can go on and download vector images as well. This is free. People make them. You can upload your own vector images if you feel like drawing your own vector images and share them with the scientific community. So go check that out as well. And if you want to draw chemical structures, MoldDraw is also a free software that you can use to create different chemical structures, and you can download them and put them into your PowerPoints as well. So those are some of the techniques and tools that I use to make presentations, etc. And I hope you find that useful. And lastly, I just want to ask if you have questions about the cake.